can't touch this.
Today I'll be presenting a piece of data collected in an experiment titled Turning a Riboflavin Binding Protein into a Self-Sufficient Monooxygenase by Cofactor Redesign. The goal of this experiment was to find a cheap and efficient way to create a monooxygenase. Monooxygenases are important because they're an organic substrate that's used as a biocatalyst in many experiments, and especially in drug treatment and other sort of biochemical research facilities. Generally, the process to make these monooxygenases needs NADPH. NADPH can be extremely expensive, and it's previously been the only way to create these monooxygenases. By cofactor redesign, the catalysts were changed, and the new catalysts are used with the original molecule and allow monooxygenases to be created more cheaply. The different molecules that they change are two separate R groups that are attached to the original molecule, as well as a flavin structure that's used to create the protein. This picture right here is of both the original form of the chemical using NADPH as a cofactor, and then they also show the way that they created the molecules cheaper by skipping the NADPH step, which allowed for molecules to be created in a more efficient and more effective transfer for the labs now. The data that I'll be presenting is from their Table 1. This table basically summarizes all the data from the experiment. It shows the 19 different entries that they used, where they basically changed R group 1, R group 2, and the flavin structure that was within each molecule. They also had positive and negative proteins, and they showed the rate of conversion from the original molecule to the molecule that they were hoping to get. The most effective combination was with the benzyl group as R1 and as the methyl group as R2, and the flavin was flavin 6, and it had a rate of 60% conversion. This was very successful, and I'm sure with more research and funding that they would find a way to possibly use this in labs and help reduce the cost of experimental drug trialing. The least effective combination was with the phenyl group as R1 and the methyl group as R2, and they used flavin 1. This only had a 14% conversion rate, and I'm sure this was an extremely disappointing result, but as I said before, they did find ways to actually make this molecule in a cheaper and more efficient way, which I'm sure they were quite happy about. In general, this experiment has allowed many researchers the opportunity to make cheaper molecules, which will eventually allow the possibility of possibly more treatments or better treatments to form, because with cheaper molecules,
You can't touch this.
to new conclusions, and with less budget restrictions because of the cheaper molecules, there's a good chance that maybe a new drug will be invented just because of a happy accident in the lab. Alright, thank you.